Certain way. How do you do that? These lifestyles, certain types of diet, when you eat, and exercise. Essentially, you want to trick your body into thinking that it's under threat of survival, adversity. You don't want it too much. You don't want to damage your body so that it can actually have the opposite effect. But you want to give it a little bit of fear, and it will have great results and give you payback for decades to come.
Okay, so I know we've talked about these other two longevity genes, but I really want, because you're the sirtuin guy, I want to dive a little deeper into what's making these things tick. And this gets a little complicated. It's a little down in the weedish, but it's important as we get to the information theory of aging and this idea that we're going to be driving toward in a minute here, which is X differentiation. So can we talk a little bit about why the sirtuins are doing what they're doing and how that works at the genetic level?
Right. All right. Well, let's do a little bit of biology. Some people haven't had a biology lesson in decades.
It's been a long time.
Yeah. Let's start with a cell. Cell is a microscopic bag of fluid packed with proteins. The proteins are encoded by the DNA, which is in a part of the cell called the nucleus. So let's become microscopic. Let's dive down into a cell and have a look what's there. So first of all, we pierce the outer membrane. There are a lot of proteins on the surface that bring in things like sugar and amino acids that are important. Now we're swimming around and we can see there's another little structure that is surrounded by a membrane called the nucleus. So let's go through there. There are little pores we can swim through.
Is what we remember from middle school biology is the brain of the cell.
It is the information, the genetic information of the cell encoded in a 6-foot long strand of a chemical called DNA.
And we're talking, this is 6 feet of DNA in this little microscopic, you can't even see it.
And there's so much of it in the body, it stretches from here to the moon and back 8 times. And it all has to be packed into very small space. And so the cell doesn't just shove this strand willy-nilly into this structure. It's not like me packing to go travel. It's actually neatly packed. And the way the cell does that is that it wraps the DNA around proteins called histones. And those histones then attract each other and eventually you get these structures we call chromosomes, which you can see sometimes with your eyes, but usually with a microscope. But it doesn't all get bundled. Otherwise, no genes could be read. Some of the genes have to be opened up and unraveled, unspooled, so that they can be turned on.
There's a really good analogy for this if you existed during the 1970s and 1980s when cassette tapes were a thing.
You might want to describe what they were.
Okay, so having existed in these times, so for the children at home, a cassette tape is a plastic cassette with a reel on either side of magnetic tape. But the important thing here for this analogy is that if you turn it on its side, only a little bit of that tape includes a lot of information, but only a little bit is readable at any given time.
And that would be a gene.
And so if your cell is trying to read your code, it's accessing just that little bit at a time. It's not accessing all of it. And that's actually what helps cells know to be cells. or the kind of cells they are.
Well, it's essential. When we're fertilized as an egg, we can be anything, but we need to have certain cell types. A brain cell has to know how to be a brain cell for 100 or more years, and a liver cell has to stay a liver cell. If you wake up tomorrow and your brain is more like a skin cell, you're in trouble. So what happens is that the packaging of the DNA is specific to the cell type, and every time that cell divides, like in a skin cell, it doesn't suddenly become a neuron because the cells inherit the information that says that gene is for skin cells, and keep that one on, but don't turn on the brain cell.
And they know this through epigenetics.
So epigenetics is the other part of the information in a cell that controls which genes are packaged tightly or unfurled or unspooled, like can happen if your cassette tape gets stuck in the machine. You remember when that used to happen?
I do, and then you'd have to take the little pencil and
wind it up. Yeah, exactly. That's part of the problem with aging, we think, is that beautiful packaging of the DNA that allows a cell to remain a skin cell forever, or brain cell essentially forever, is lost. You start to get this unspooling of the DNA, and genes that shouldn't be on start to come on over time.
Genes that shouldn't be readable are now readable, and so you have like all this extra information floating out there that the cells can read, and because the cells are now reading large parts of the code instead of the very specific parts of the code, they get confused?
Right. And we call it X differentiation. Differentiation is the common term for cells becoming a certain cell type. And we've coined the term X differentiation, which is essentially cellular confusion. They become more of a generalized cell type rather than a specific one, because genes that shouldn't be on start to come on over time. And that, we think, is a root cause, if not the root cause, of aging itself.
Let's unpack that a little bit. Because, it is kind of a pun. The thing that you've told me, and that really just like sort of completely shifted the way that I was thinking about aging, even after actually working with you for quite a bit, is that this is not just the root cause of aging. You believe that X differentiation is the root cause of many, if not most, diseases?
I don't just believe it. I think it's now a fact. When you look at Alzheimer's disease, and unfortunately up until the last few years, we were treating it at the end stage of the process of aging, which results in plaques and tangles and dementia. But the process that led to that disease was happening from birth, and that's aging. So Alzheimer's, just to take one example, is an age-related disease that's not just age-related, it's actually 90% of it is caused by the aging process, this unspooling, this X differentiation. If we were able to slow that down, you'd have Alzheimer's much later. And now we're showing in my lab, and we'll talk more about this, but I want to mention it because it's so profound. We can reverse the age of a brain in an animal. And when we do that, we're showing, and we will continue to test this, that those diseases go away, proving that those diseases are actually caused by aging.
Okay, so let me see if I can put this into sort of some kind of a picture. We have these brain cells.
Neurons, yeah.
Neurons. We have neurons. Over time, because our DNA unspools, it becomes less tightly packed. The code is more readable. For instance, the code that makes a liver or a kidney. And so that brain cell is sort of also part kidney cell?
Right, and we can read which genes are on. We call it gene expression patterns. It's very easy.
So you actually see this in Alzheimer's brains. You can see that the cell is
losing its identity.
Right,
losing its mind, so to speak. And we can see that with normal aging too. Even if you don't have Alzheimer's, during aging, I'm 50 now, so some of my brain cells are undoubtedly getting confused and starting to believe that they're part skin cell, part kidney cell, and that's probably the reason why even in your 50s you cannot function like you did when you were 20.
Okay, that's dementia, that's Alzheimer's. What about other diseases? There's another one that's a good example for this, type 2 diabetes.
Right, high blood sugar.
Same process.
Yeah.
Same problem.
Well, it is.
These aren't two different diseases. These are the same thing.
Well, that's a major point, that we've been looking at diseases as separate things. This is how modern medicine works. We need a different drug for type 2 diabetes right now, and for Alzheimer's, for cancer, for frailty. muscle wasting, bone loss. I'm saying that the same process is leading to all of these diseases, and if we can slow down, and even now we have a technology to reverse that process, those diseases will be slowed down and even go away by age reversal.
Okay, I'm on the cusp of this. I want you to spell it out for me though with the diabetes, and then I want you to do like, I like things in triplicate. So I'm convinced on the Alzheimer's. Explain what's happening in the diabetes that makes it the same process.
Yeah, well, so type 2 diabetes, in part, is a problem of bringing glucose, sugar, out of the bloodstream into your muscles and your brain, which are the major sources or major requirement is glucose. And if you don't suck the sugar out of your bloodstream, you're going to have basically treacle or caramelized protein in your system, which is going to cause all sorts of havoc.
Sounds delicious, but it's really a bad thing.
No, you don't want to have glucose attached to proteins. In fact, you can measure it. Doctors, when they do a test called HbA1c, that really means how much glucose is physically bound to your hemoglobin, which carries your oxygen. And it's a percentage. So if you have over 5.7% of your hemoglobin attached to glucose, you're pre-diabetic. And over 6.5, I believe the number is, that you're actually type 2 diabetic at that point, and then you go on metformin. The point being is that having high levels of blood sugar, we know, predisposes you to other diseases, heart disease, frailty, dementia. You don't want to have a lot of sugar in your blood.